Welcome back. We are now moving into our second to last chapter, chapter 16, which is the regulation of gene expression. I think the textbook calls it genes and how they work, but what we're really looking at is how an organism controls whether or not it's expressing a certain gene. Because as we'll see in many different examples, there are times when you may want to activate a gene because a particular resource is available to you that you need the enzymes for to break down. And there are other times when you don't want a gene to be active because it would be a waste to produce the enzymes that are associated with that gene if you don't have any use for them at the moment. Or, another example would be there are times when certain genes should be activated for growth and cell division, other times when you don't want to have those genes activated. So we're going to look at the general characteristics of controlling gene expression, mostly in prokaryotes, but we're also going to spend a little bit of time talking about eukaryotic gene expression. So we'll first introduce the concept of regulatory proteins, which are going to be proteins that are associated with regulating what genes are being expressed at a given time. And then we're going to take a look at a couple of specific examples of prokaryotic regulation, specifically the lac operon and the trip operon, or the TRP operon. And if you think back to when we were talking about gene clusters, an operon in prokaryotes is a cluster of interrelated genes that are all going to be transcribed at the same time. So they're all, for example, enzymes that are going to be involved in a biological pathway. And this way, if one of them is being transcribed and translated, then the rest of the genes that are nearby will also be transcribed and translated, which makes a lot of sense because if you're dealing with an enzymatic pathway, in the case of lactose, for example, which is going to take you from lactose through a series of intermediates to glucose eventually, then you definitely want to have all of the enzymes necessary in the cell. And so clustering them together enables you to do that. Eukaryotic regulation is really quite different though, and we don't have what we call operons, but we do sometimes have clustered genes, but we'll talk more about that in a moment. Then we're going to look at how chromatin structure affects gene expression. If you recall, DNA is wound up around proteins called histone proteins, and depending on what segments of DNA are wound up around those proteins and what segments of DNA are exposed to the environment, you have variation in what genes can be transcribed. So the structure and the shape of the chromatin, the combination of DNA and protein, actually determines what genes can be transcribed at a given time. And if, for example, your genetic material for a gene is wound up around a histone, then the enzymes that are necessary for transcription and translation can't access that DNA, and therefore the gene won't be transcribed and translated. So structure of chromatin plays a huge role in the expression of eukaryotic genes. We'll briefly look at a couple examples of post-transcriptional regulation, although we've already kind of talked about this. If you recall, after transcription, the mRNA is going to have a 5' GPT cap, a 3' poly A tail, and a variable splicing process that it undergoes. So this variable splicing or alternate splicing can result in many different products from a single mRNA, and this is one way to regulate genes as well. We're not going to talk about protein degradation in this chapter. We're going to leave that out. So gene expression is necessarily controlled for developing and growing organisms because there are times in an organism's lifespan that it requires certain things, and those come from, they have a genetic basis, and there are times when those things are no longer necessary, right? Or perhaps there are times when a gene that only is activated in adulthood or maturity of the organism is not active at a younger age. So we use a lot of different mechanisms to control gene expression, but one of them is a regulatory protein. And these are proteins that are going to bind DNA, and they modulate the ability of mRNA, or I'm sorry, RNA polymerase to bind to promoter regions, or regions where RNA polymerase binds. Remember, we talked about this in the last chapter. There are segments of DNA that RNA polymerase will attach to called promoters, and from there RNA polymerase will proceed to read DNA and produce an mRNA template. So eukaryotic cells are not as responsive to their environment in terms of their changes in DNA transcription and translation or gene regulation as prokaryotes are. Eukaryotes have a pretty well-established plan for their lifespan. Prokaryotes are at the whim of their environment a lot more. You've got to think about it this way. If you're a prokaryote and you're a single-celled organism and you don't respond to your environment immediately, the one cell that you are is incredibly vulnerable. In the case of a eukaryotic cell, we can lose hundreds or millions of cells in a single day and be fine without any issues. So it's not as important that our cells adapt and respond extremely rapidly to the external environment, but instead that they're constantly maintaining a constant internal environment. So gene expression, again, is going to be controlled by regulatory proteins that bind to DNA sequences. These regulatory proteins are going to attach to what we call the major groove of DNA, and we'll see a picture of what that means in just a moment, but essentially what we're talking about is the wider groove of DNA. If you remember looking at pictures of DNA, you may have noticed that there's a little bit of asymmetry in terms of the distance between the phosphodiester backbones in the two openings or grooves of DNA, and the major groove is the larger one. The size disparity is what we use to distinguish the two from one another. So if you look at DNA from top down, you can see that there's a little bit more space between the nucleotides on the major groove than there is on the minor groove. And this is because of the orientation of the phosphate group in the nucleotides. They both sort of point about 45 degrees away from the center of the groove, or the center of the circle. If the center is right here, they're about 45, not quite 45, but something close to 45, maybe like 70 degrees or something like that. So if this one is pointing 70 degrees away from the center, then I guess this one would be 310. I don't know. It's been a long time geometry, so someone might have a laugh at that. But anyway, I think it would be 310 because this should be 360 and 0, right? So 70. Yeah. Anyway, the point is, because of the orientation of the phosphate group, you're going to see that there's a smaller gap between the nucleotides over here than there is over here. There's a lot more space over here. And what that creates is a larger groove right here and a smaller groove right here. It's more narrow between the phosphodiester backbones here than it is here. And that's the difference between the major and the minor groove. So in prokaryotes, transcription can be regulated in a number of different ways. We refer to positive control, which is going to increase the frequency of transcription and translation of a particular gene, or negative control, which is going to decrease the frequency of transcription and translation. So we associate positive control with regions of DNA called activators, which are going to encourage RNA polymerase to bind to a promoter and transcribe the genetic material. Negative control is associated with what we call repressors, and repressors are molecules that are going to bind to activators or promoters or regions between them called operators that are going to prevent the RNA polymerase from binding to the DNA and transcribing. So prokaryotic cells, again, have to respond to their environment rapidly. And when they do this, we distinguish between induction, which is the initiation of the production of a set of enzymes for a chemical pathway when they find the substrate for those enzymes in their environment. So the first example that we're going to be looking at is the enzyme group that is going to convert lactose into glucose through a series of intermediates. Now, the substrate for those enzymes is lactose. The enzymes themselves are lactases and a few other intermediates. Now, by contrast, there's also repression, which is going to mean that the cell, even though it's capable of making a particular enzyme, is going to be prevented from making that enzyme for some reason or another. And we're going to look at this in the TRP operon. So the lac operon, as I was mentioning before, is going to be the class of enzymes B-galactosidase, permease, and transacetylase, which are LAC-Z, LAC-Y, and LAC-A respectively, are going to be the enzymes that are necessary for converting lactose into a usable energy source in cellular respiration. So the genes for the LAC repressor are on the LAC-L gene or the LAC-L region, and this LAC repressor is actually associated with the rest of the operon. And you might be thinking, well, why would you want to transcribe a repressor when you're trying to transcribe the enzymes necessary for breaking down lactose? And you'll see why in just a moment. So this is what the LAC region of an E. coli chromosome looks like. There's a promoter where RNA polymerase can attach. There are genes for transcribing the repressor protein. And the promoter is just upstream from the repressor. So if RNA polymerase were to attach here, it would travel in this direction. It would produce the repressor protein indirectly, but eventually. And the repressor protein would then bind to the operator. But in certain circumstances, as we're going to see in a moment, this repressor system gets shut down and the repressor protein gets removed from the operator, meaning that RNA polymerase can attach to this promoter and transcribe gene Z, gene Y, and gene A for B-galactosidase, permease, and transacetylase. Now, normally the lac operon is turned off because lactose isn't always going to be in the environment, and prokaryotes don't want to waste their resources producing enzymes that they can't make use of. So they hold off until the evidence of lactose in the environment appears. So the lac operon is shut down by this repressor and blocked, then transcription is blocked. But when lactose is present, lactose gets converted into allolactose, and allolactose is going to attach to that repressor protein. When allolactose attaches to that repressor protein, it changes the shape of that repressor protein, and it can no longer attach to the operator, which was what was preventing transcription of the lac operon in the first place. And this permits transcription to proceed. So this is what that looks like. You have RNA polymerase, which is going to be attaching itself to the lac repressor or the lac operator and producing the lac repressor gene ordinarily. And that lac repressor protein is going to attach to the operator of the rest of the lac operon, and this is going to prevent the RNA polymerase from attaching to this operator and transcribing those genes. However, when lactose shows up in the environment, it binds to this repressor protein, which opens up the operator, and RNA polymerase can attach to that operator and transcribe genes Z, Y, and A. So there's also what we call glucose repression, which is involved in the process of regulating the lac operon. Glucose repression is a way for the cell to preferentially use glucose when other sugars are present. So when lactose is present, the prokaryotic cell might initially think, oh, okay, if lactose is here, I should start transcribing the lactose family of enzymes, the enzymes that we need to break down the lactose. But if there's enough glucose in the environment, then the cell would rather focus on glucose because it can do glucose cellular respiration more efficiently. So there's an activator protein, the catabolic activator protein, CAP, which is going to attach to the lac-operon and prevent the transcription of the lac-operon by promoting the transcription of the lac repressor, and we'll see that in just a second. So you see here, when CAMP is present, that means that glucose is going to be low. So CAMP attaches to CAP, and that attaches CAP to the DNA of the lac-operon, which leaves the lac repressor open. So the RNA polymerase, or I'm sorry, not lac repressor, the lac operator open, which means RNA polymerase can attach and transcribe. If glucose is high, however, RNA polymerase is not going to be able to transcribe these because the repressor protein is going to remain bound to DNA. And this is because the high glucose is going to eliminate CAMP in the cell, which is not going to then bind to the CAP protein, and the CAP protein cannot bind to the DNA. So in effect, what's going on here is when glucose is present, then CAMP is in low concentration. When CAMP is in low concentration, CAP does not change the transcription of this lac-operon, which implies that the lac-operon will not be transcribed. But when CAMP is present, that indicates that glucose is low, and that means that it's worthwhile for the cell to produce the enzymes associated with lactose, and so the CAMP binds to the CAP. CAP then is the appropriate shape to bind to the lac-operon, and this promotes the activity of RNA polymerase on the lac-operon. So another example of an operon in the prokaryotic systems is the TRP operon, or the tryptophan operon, which is an essential amino acid, or a necessary, I shouldn't say essential, a necessary amino acid in prokaryotes and also in eukaryotes. Now, ordinarily, the operon is always running, but if there's enough tryptophan in the cell, the operon will shut down, or in other words, the enzymes necessary for producing tryptophan will no longer be transcribed. And this makes sense, because if you have enough tryptophan in the cell, why would you want to produce more? It's a feedback mechanism that when you produce enough tryptophan, it shuts down earlier stages of the process. So it's negatively regulated by the TRP repressor protein. And the TRP repressor protein is going to, much like the lac repressor, bind to an operator and prevent transcription to take place. Now, if tryptophan levels are low, tryptophan is not binding to the repressor protein, and therefore the repressor is unable to bind to the operator. And this is because the repressor itself requires tryptophan in order to bind, and you'll see that in just a moment. So depicted here is the inactive TRP repressor. This repressor has a morphology or a conformation or shape that will not attach to DNA ordinarily. But if there is tryptophan in the cell, the tryptophan is going to bind to this repressor protein, changing its shape, and it can then bind to the operator to prevent transcription and translation. But when there is no tryptophan in the cell, the repressor protein cannot bind to the DNA, and so the RNA polymerase is able to transcribe the genes necessary for tryptophan synthesis. Now, eventually, after transcribing these genes and translating these genes, you're going to have all the enzymes necessary for producing tryptophan, and therefore tryptophan is going to be produced in the cell. As tryptophan accumulates, it begins attaching to these repressors. These repressors change their shape in response to the binding of tryptophan, and they attach to the operator of the operon. And this prevents RNA polymerase from transcribing the operon, and therefore it halts the production of tryptophan. So prokaryotic regulation works in these operons, but eukaryotic regulation is really quite different for a number of reasons. First of all, in eukaryotic transcription, there's a lot more complexity to their regulation. It's not as simple as a single repressor binding to an operator site and preventing the process of transcription, but instead you'll have many different promoters that are going to be involved in encouraging the formation of a transcription complex that is going to proceed to transcribe a gene once all of the parts are in place. There's also the fact that eukaryotes have a more complicated arrangement for their storage of DNA. Remember, prokaryotes don't utilize chromatin. They just have their DNA loosely associated. They don't have it wound up like beads on a string the way that eukaryotes do. So eukaryotes have a few mechanisms that involve the tightening and loosening of these chromatin structures in order to prevent or permit transcription. So there are what we call general transcription factors in eukaryotic transcription, and these are necessary for the assembly of the transcription apparatus at a particular site on the gene. Now, or DNA, I should say. When you have the general transcription factors necessary for transcription, they will promote RNA polymerase to bind to a promoter, and then RNA polymerase can proceed with transcription. Here are a slew of the different proteins involved in the general transcription initiation complex. There are A subunits of the protein, TF2D, H, E, F, TAFs, Bs, lots of different proteins that are associated with the initiation complex. RNA polymerase II is not going to bind to the promoter until all of these transcription factors are in place. So by regulating the quantity of any of these transcription factors, you can promote or prevent the transcription of genes. Now, there are also what we call specific transcription factors, and these tend to be more tissue specific. So for example, there will be specific transcription factors that are active in nervous tissue or brain tissue that wouldn't be active in your epithelial tissue of your stomach. And this is because there are certain genes that we want to activate in some cells and other genes that we don't want to activate in those cells. So chromatin structure is another mechanism by which eukaryotes regulate DNA transcription and translation. DNA, remember, is twisted up around these histone proteins, and these histone proteins can be tightened or loosened in order to expose or restrict exposure of DNA. There are also what we call epigenetic alterations, and these are alterations in chromosome structure that don't have anything to do with changes in the DNA sequence. They are heritable, so they can be passed from one parent to another, but they are not the result of mutation or changes in DNA sequence the way that most heritable characteristics are. Now, chromatin modifications can include epigenetic, or epigenetic inheritance can include chromatin modification, specifically types of DNA methylation. So DNA methylation is when methyl groups, or DNA methylation is when methyl groups are attached to a certain region of a chromosome, and this inactivates or deactivates the genes within that region because it changes the shape of the chromatin in those regions. Another good example of chromatin modifications that result in epigenetic inheritance is X chromosome inactivation. If you recall, we talked about how female individuals, or assigned female at birth individuals, have two X chromosomes, but they shut one down. They condense it into a structure called a bar body, and it doesn't get activated. This is a chromatin modification. They tightly pack up that X chromosome so that it doesn't get utilized. There are also what we call histone modifications, and there are 4 possible modifications for histones. All of them have different effects on transcription and translation. Acetylation, methylation, and phosphorylation are the most common of them. And when you acetylate a nucleosome, or a histone tail, it's going to change the shape of the chromatin such that the histone unwinds the DNA, and that makes the DNA in that region more accessible. So you go from a condensed solenoid, or a condensed cluster of DNA wrapped around histones. You add acetyl groups to the tails of these histones, and that causes a morphological or chemical change in the histone, which unwinds the DNA and exposes regions that can be transcribed. There's also what we call post-transcriptional regulation, which is where gene expression is controlled after transcription takes place. Everything we've talked about up until this point was a way of regulating whether or not transcription would take place. And so there are a number of different mechanisms that take place after transcription has already taken place, including some of the RNAs that we already talked about. The small RNAs like microRNAs or small interference RNAs that are going to break down certain mRNAs before they can be translated. There's also what we call alternative splicing. If you guys recall, when eukaryotic chromosomes or mRNA are spliced, you're removing the introns and rejoining the exons. And you can eliminate some of those exons, or include all of the exons in different orders in order to get different protein products. There's also RNA editing and mRNA degradation, but we're not really going to talk about those.